you and everyone. Today, me and my group are going to present our entomology collection report. Our group consists of Farheen Nayaab, which is me, and my friend MAAHIN IBRAHIM. Now, MAAHIN will continue with the presentation.
My name is MAAHIN and I'll be giving a brief introduction about arthropods. Arthropods are invertebrates with jointed legs. The main characteristics of arthropods include having an exoskeleton, segmented body, bilaterally symmetrical open circulatory system, sexual reproduction, ventral nerve cord, jointed appendages. Arthropods consist of the following subphylums: Hexapoda, Meropoda, Chelicerata, and Crustacea. Our insects were captured in various places like parks, residential areas, and fields.
Objectives of this report were to collect and pin members of the phylum Arthropoda, to label insect body parts, as well as identifying and classifying them according to the order, family, and species. Another objective was also to identify and apply the correct technique in insect collection and preservation. Various materials were used to catch and kill these insects. Some of them were chloroform, forceps, DIY fly trap, gloves and sanitizer with 70% alcohol, foam, pins, and an airtight bag.
Some insects were caught with the help of insect repellents and were kept in the freezer to kill them. Others were caught by hand and by the help of DIY traps. Some of them were preserved by using alcohol and other chemical agents. After freezing them, they were adjusted properly so that all the appendages can be seen and counted. Then they were pinned on a foam block and photographed. Our insect collection consists of 4 insects in total. Figure 1A is a yellow spotted millipede, which was caught by me in a park. And figure 1B shows an ensign wasp, which was also caught by me in my house. Figure 2A is a winged carpenter ant. This was caught by Farheen and the collection site was a field. Figure 2B is a housefly. It was also caught by Farheen and the collection site is her house.
Life cycle of our insects. Now I'll be talking about the life cycle of the insect that we captured. Some of the insects undergo incomplete life cycle, which means they are hemimetabolous, and some of them undergo complete life cycle, which means they are holometabolous. This is the life cycle of a millipede, which undergoes incomplete metamorphosis. This is the life cycle of a housefly, an adult wasp, and an ant, which all undergo complete metamorphosis. The techniques that we use to capture these insects include making a DIY fly trap, which was made at home, a freezer, which was used to kill the insects, a repellent, which was also used to kill the insects. The freezer was also used to preserve these insects. Some of the insects were caught by hand.
Out of the 4 insects that we collected, 80% of them were from the class Insecta and 20% was from the class Diplopoda, which was the millipede I caught. Regarding the subphylums, 80.6% of insects were from subphylum Hexapoda and 20.2% from the subphylum Meropoda. Life cycle, here the shaded region represents the insects that undergo incomplete metamorphosis and complete metamorphosis. A total of 3 insects undergo complete metamorphosis or they are holometabolous, and 1 insect undergoes incomplete metamorphosis or they are hemimetabolous. In conclusion, insect collection is an important method of studying insects and classifying them further. This method is especially beneficial to humans as dangerous diseases and their vectors can also be studied along with the insect, which provides important information on how to stay protected from those diseases. It also provides important historic data revealing relationships of different insects with each other. The objectives of this report were met with 4 insects collected, pinned, and labeled with the common name, site of collection, phylum, subphylum, class, order, family, genus, and species name.
These are the references which were used to complete our project, and this is the task assignment. To sum up, this project was an amazing experience as we could learn how to catch and preserve insects. We could learn how to apply hands-on what we had learned in our entomology collection. This way, all the objectives of this project was met. That's it from us. Thank you.